Thank you for the invitation to be here. I'm very excited. I think last time I was here was 2009, visiting the department, so it's very good to be back. What I'm going to try to do here is go from a very macro aspect or macro view of the topic, and then hopefully try to get to some more of the basics or some of the things that I believe we should be learning more or should be trying to understand better, but also some aspects that I think we already learned and we can implement already in dairy farms. So that's kind of, I'm as an assistant professor, I was hired as a extension with an extension appointment and teaching appointment, but I was given a lab and say, we need you to do research as well. So in Illinois, our extension is pretty much applied research. That is the expectation. So that's kind of the things that we are trying to do. Go to farms, get some information, and do some research, and collaborate with people. Some of the data I collaborated even with Anna when she was a PhD student in Florida, for example, with Dr. Peter Hansen. So we're going to talk a little bit about it. One of the things that we usually, when we are talking to farmers or other audiences, and we ask people, especially during the transition period for cows, what do they want from those cows? And usually everybody get it right. Everybody say, hey, I want cows to produce a lot of milk, that they don't get sick, and they get bred, and with that I'm gonna make a lot of money. So it's easy. If you go out there and ask anybody working in the dairy industry, that's what they want from cows. So that's some kind of the things that we're gonna try to match here. This is a draft or a summary with all the kind of metabolic disorders happening around transition period. So this is a table put together by Zoetis, and it just went there out in the literature. So there are more than 9 research papers here, and they just put, hey, this is what we are finding. How much does it cost each of those cases? So we had some data in specific papers. You just put all together. Hey, this is it. If these cows get sick, they're gonna get more chances of getting out of the herd. That's what is the message here. One of the things that everybody identified more than 20 years ago is that this period, 3 weeks before, 3 weeks after calving, is the most challenge for the cow, and she gets some of these disorders. And why is that? One of the things to highlight here, I'm putting the requirements for a cow 2 days before calving and 2 days after calving. So I'm talking about a cow and a heifer. This is roughly how much energy she would need before calving. This is how much energy she would need after calving. 2 days. I mean, I'm talking about the span of 4 days. And usually, we can easily make cows eat this much before calving. However, after calving, we all know that is very challenging. So you go through that negative energy balance period. She has to take some out of her body, adipose tissue, to try to get some energy going on her body to try to fulfill this, but it is still very hard for cows to do that. One of the things that we can see here that we always look at is the metabolizable energy 7 days after calving. So here I'm showing you, this is the requirement for that cow. This is how much usually cows can eat. So usually 70% she can get it. And this is the percentage going just to lactation, the percentage of what she can eat. So a lot of what's happening here is that she needs to fulfill that lactation, that milk production. So that's what's happening. A lot of that energy is going there. One thing that sometimes we forget is also the intake is reduced, but also metabolizable protein, it's going to be the same story, This is the requirement for the cow. This is how much she usually can eat to fulfill the requirement. And this is how much is going to lactation. So that's the story. We have this cow that is not being able to eat as much as she needs, but now we can see this metabolizable energy, metabolizable protein, all that getting to the intestine of the cow. Another concept, and here I'm going to try to bridge those, the nutrition part with the reproduction cow part of it. We know that cows are producing more and more milk every time, and conception rate has been decreasing and everything else. Perhaps at some point here, and maybe I was even talking to Anna that I need to update this and see what's going on until 2015 or something, But we did a lot of improvement, especially in the bull selection, and talking about, hey, we should look at fertility, daughter pregnancy rate, to try to pick it up a little bit of this drop in conception rate. And I think we've been doing a better job on that, especially in 2009. But this is one of the things that is happening. One thing generated from data here from California, that we believe that milk production or high yield is not associated with reproduction. So what I'm saying here, for example, this, you have 4 TMR herds, and what was done is just divide in quartiles the average, the milk average. So for example, you have cows producing 32 kilos per day or 50 kilos per day, and when you look at cyclicity by 65 days, so if they're coming in heat or not, you can see that if something, perhaps cows that are even producing more milk, they're cycling more than cows producing less milk. So it doesn't seem to be associated farms, at least in a farm environment, farms where cows produce more milk, doesn't mean that the cows are going to be with poor reproduction. And perhaps this also brings a lot of management things. So I think it's a lot of things are confounded here. And the same thing with pregnancy rates. So high milk production doesn't seem to be detrimental to reproduction when you look at this data. One other thing that really exists in the farms, and when I put together a bunch of trials here with a bunch of cows, and I look at, hey, let's preg check this cow at 28, 30 days, and then let's look at 40 days later. And you're going to see that this pregnancy loss is always present, always. And we can go from 10, 9, until 20%. So let's say 15% is something very normal. And that's something that we see out there all the time. Of course, farms sometimes they don't do all this checking and everything else, so some farmers may not even know about it. But that's something that we have, and we think that we may be able to impact that in a positive way. The other thing that we can look at is that pregnancy loss is one of the numbers that we check, but however, throughout the whole cycle, since the cows are bred, we know that failure in conception or in fertilization can happen. So pretty much whenever you have 100 cows, you're going to end up with only 33 of them getting really pregnant and giving a calf to the world, let's say. Make sense? So we can talk about, different aspects, and I know different, people study different stages of that, but the idea is I had so much work getting this cow pregnant, and now 15% of them are going to lose. So that's where we can impact that. Why is that important for farmers? We know that getting cows pregnant return to the pocket of the farmer. So this is a tool actually from the University of Wisconsin, Victor Cabrera. He has that. You guys can go there and you input all the data from the farm. So the cost of production, price of milk, everything else, and it's going to tell you, for example, the net return by how fast can you get your cow pregnant. So your net return is much higher sooner you can get that cow pregnant. And this is just a curve and say, hey, with time, cows are going to get pregnant and pregnant. But that's why a lot of this synchronization protocols, farmers are so eager to do it because they understand this. They know that getting a cow pregnant soon returns that into money for them. So that's why they become so popular and so they are so eager to do that. And not in a linear way, but as we increase pregnancy rate, the money that returns per cow per year is going to be higher and higher. Not in a linear. So of course, if you're going from 25 to 30, it's much less impactful in your pocket than if you're going from 16 to 20, for example. Average in the US, 17%. That's our average. 17% preg rate. I believe that a really standard goal would be 20% for farmers out there. Top herds, they can get 30% preg rate all year long. So if you see Hordes Dairyman, they post some of the best herds in the US, and you can see that most of them, they are 30% preg rate. So it's possible. one of other thing is that we need to be aware of it, that all these diseases that I've mentioned before, they're always going to be impacting reproduction. So this is work done by Ribeiro, and what they saw is that they got clinical disease, so it would be ketosis, milk fever, they didn't see much. But whenever I have one disease or multiple disease associated, like mastitis, your conception rate now, your pregnancy would be reduced when I compare to cows that didn't have. And even the subclinical, so high levels of NIFA or high levels, serum levels of BHB, you'd see that just one alone would not cause that, but two or more then would cause a reduction in that. So we need to be aware that anything that we are also doing that reduces those diseases or disorders are going to be helping those cows to get pregnant. This is a pretty interesting chart that tells you 7 factors that are going to affect pregnancy in cows. All these are going to be affecting pregnancy. And today I'm going to be talking about 3 of this. So body condition score or postpartum uterine diseases, talking about the quality of the oocyte that is going to match that semen to create the embryo, and also the embryo itself. And one of the things that we have to remember that by 14, 18 days, that embryo is going to be attaching the uterine wall. And one thing that is going to tell the cow, hey, don't come in heat again, is going to be this little thing here, interferon tau. And that is directly associated with the size of that embryo. So if I have a bigger embryo, I can have more of that interferon being transmitted to the cow, signaling to the cow, and that may be helpful for her pregnancy status. So I want you guys to remember here, because this is going to come back to the end of the presentation, actually. Just a quick review, and I'm going to start getting now into the protein side of things. And pretty much what I want to highlight is we are going to be feeding cows. They're going to be eating, and we are still challenging or still using the term crude protein. So that's the amount we're going to have in the diet. We're going to have something that is going to be degraded by that's going to be degraded by the rumen in producing bacterial protein. We're going to call that RDP, or rumen degradable protein. Part of that protein is going to go straight to the duodenum, going to be absorbed. We're going to call that RUP, right? So that's kind of the 2 things. And one of the things that we have, whenever we are degrading that protein, you have this release of that N component, or you're going to have a lot of the nitrogen being recycled by the cow as well. So as I feed more protein, I should have more of that nitrogen being recycled by the cow. So that's kind of overview. RDP, RUP, whenever I have the combination of that, what the cow is going to be using is that what I said in the beginning, the metabolizable protein. That's what the cow is going to be getting into her system. One thing that I want to convince you guys here or talk to you is that this amount of protein is going to be showing up in the milk, for example. And that's one of the ways we can assess that or that excess. So you can see here research talking about, hey, if I feed more RDP, RUP more than what the cow needs, I'm going to start having more of that nitrogen showing up in the milk. So if you guys feed a lot of protein to cows, that's going to show up in the milk as MUN, nuclear nitrogen. I can also build a link between the amount of nitrogen in the milk with the amount of nitrogen in the plasma of that cow. So usually that's going to reflect a direct association between those 2. And what I have here is putting together 2 trials where I'm putting the plasma of the cows, and what we see here is pregnancy per AI. Whenever I have more or less than 19 milligrams per deciliter, or less than 19, and you can see there is a huge difference. So with this data, it seems like if I have a lot of nitrogen circulating through the cow or showing up in plasma and milk, I'm going to have, if I have a lot of that, I'm going to have lower pregnancy rate. Make sense? So why would that be happening? Why a lot of nitrogen or a lot of protein would be bothering that? And one of the things that we have out there is from DHI data, a group from from Maine, what they did was, hey, let's look at the MUN of cows in the milk, so urea nitrogen in the milk, and let's associate it with reproduction. And this is what they saw. I mean, that's a huge paper, more than 10,000 cows on that data set. I'm going to summarize to get you to the point that I would like to share with you is one thing that is we have to kind of be looking at is what is the level of production or how much milk that cow is producing. So what they compare, and they do spring and fall here. I'm just going to give an example in the spring. So if I have a high milk production cow, that's how they classify it, and that would be 113 pounds per day, and she has 20 milligrams per deciliter of MUN. This is going to be her first AI conception rate. So let's see 35%. Now, if I have a cow with the same amount of milk urea nitrogen, 20, but now she is a low milk production one, classified as low. And you can see that her conception rate, it is 40%. So one thing that we need to be sensitive is, depending on the production of the cow, the MUN may be good or bad. So sometimes in Europe, they always mention, hey, our cows here, they have higher MUNs than your cows there. We're going to talk about, you guys should have between 8 and 12 MUNs here in the US. Perhaps in Europe, in a grass-based system especially, that should be different. And I think it comes back to the point of, hey, those cows there, they're not producing this much milk, or cows in Argentina or Brazil. So we need to think a little bit different. But also what may be happening on those two factors impacting reproduction can be explained maybe by this trial here. Very interesting stuff. They got cows and they put either saline in the blood of the cows, and they were able to measure the uterine pH and also the plasma urea nitrogen. So whenever I put saline, that's what's happening. But also another treatment, they put urea to be similar to that high nitrogen going up. So what happened here, whenever I injected the urea treatment into those cows, as expected, the plasma urea nitrogen went up, right? But look what happened with the uterine pH. That uterine pH went down. So that's one of the factors that can be contributing to that poor reproduction whenever you feed a lot of protein, or when you feed, or we have a lot of nitrogen. Whenever you do that with the saline, nothing happened. And there is also some other data from in vitro talking about prostaglandin production that is affected, and that may be impacting as well reproduction on an in vitro system. So that's kind of why we think feeding high protein is not a very good idea. And we measure that by this high nitrogen in the milk or in the plasma. We have to remember here that proteins actually are built off amino acids, right? And this is something that I got from Illinois here. Don't talk about essential from Dr. Baker's point of view. We talk about non-dispensable and dispensable amino acids, not essential, non-essential. All amino acids are essential. That's what he used to say. Some of them we can dispense, some of them we can't. So we need to get these ones here from the diets. So this is kind of the idea of what are amino acids. We think about proteins, but actually amino acids are building those proteins. And just to give you an idea, this is a project that we did 2 summers ago. So we sent letters to all the farms, and we selected 20 farms to visit. So with a team of students, grad students and undergrads, we visited 20 farms in Illinois. And we collected the TMR samples from those farms, and we evaluated for crude protein, for example. This was the average, but look at the range. So here's something very funny, because sometimes the average always going to look good, but when you look to the range, then that's when you catch stuff. So I remember a extension agent talking about it, and was very, highlighted to me how important it is, the range. So she said, hey, if I'm in the kitchen and I put one arm in the freezer, the other arm in the oven, on an average, I'm pretty good. However, on those extremes, I'm pretty messed up, right? So we have from 20 to 21% crude protein in this TMR for lactating cows. So what I did was, hey, let's see an average cow, Okay? So I put all the requirements, and you go to NRC, and it's going to tell you, hey, this cow usually needs 15% crude protein. And this is the samples from all the farms. So this is I use as well as a benchmark tool to provoke change. So what happens? Every farm that I visited, they got a number. They are the only ones that know the number. Nobody else knows. So I'm presenting this, and they are looking their farm. Oh, crap, I'm here. Look, my neighbors. So I divided Central, North, and South Illinois. So I think this is something that really causes change when you see your neighbor is getting something and you are not. Then farmers get crazy. They really want to change. But some people may be underfeeding, but most of the people, they are overfeeding that crude protein. Again, we already talked about some of the impact, and I don't even mention that, crude protein or protein usually it's expensive. So farmers, they have to pay for it. How about amino acids in reproduction? We know that embryo, the placenta development, it's needed there. However, there are a lot of other things that those amino acids are responsible for. So what I want to get to you guys is that protein or amino acids for milk protein is not just that. There's other things going on in the body of the cow that those amino acids, specific amino acids, are needed. We know that these two here are the most limiting amino acids for dairy cows, methionine and lysine. This is an average, so this is how much you have in milk of those amino acids. And here is an average or mean for the bacterial protein. So it's a very good source of protein for the cow, the bacterial protein. If you can get as much as you can, we should go for it. Get as much as we can. But however, when you go to other common sources, corn or soy, you can see that for those two amino acids, it's pretty hard to get a high amount of it. And maybe that's one of the reasons why they are sometimes limiting as well. It's hard for us to feed enough of that. I've mentioned to you guys those pregnancy losses. So now what I'm trying to say, can we, with methionine, the most limiting amino acid, prevent that? Let's see one thing. This is a very interesting paper, okay, that what they are trying to do here, develop rat embryos in a media that has cow serum. So I think it comes back to our kind of niche here. We come back to the cows, even though it's rat embryos. And this is a table. What they did is, I'm putting cow serum, and I'm letting that rat embryo to develop. If I just put the cow serum, this is the amount of protein that I'm measuring from that embryo, and this is the percentage of abnormal embryos. So 100% of the embryos, they were abnormal. Now they did a little bit different. They add amino acids plus vitamins to that serum. Now, look at the amount of protein, 0 abnormal. And they start playing with that. Hey, how about just amino acids? Boom, 0. How about just vitamins? Nope, we are back to the abnormal stuff, 100%. How about amino acids without methionine? All of them abnormal. Now, how about just methionine then? And that's what they got. So you see that whenever you have methionine, that embryo was able to develop to normality. So one highlight, hey, methionine is very important for embryo development. One of the things that we started talking about, and we know a lot of the nutrition impact on the embryo and in the offspring, even from different species and even from humans. But when is the best moment? When is it really needed? That's kind of our question. So in a farmer perspective, okay, I need to feed methionine to my cows. When is the most important period that I should do it? We come back to things like, for example, the primordial follicles or the preantral follicles, and that's the ones that we are going to be breeding long time ago. So if I want to breed a cow at 70 days after 70 days in milk, probably I should have done something in the prepartum period. So I can have that follicle come up in an environment that is already high in methionine. But again, this is all in my head. We don't have a research on that yet. So we don't have all those answers yet. But I think we should be looking a lot on these things here. That's why heat stress, and you guys here do a lot of stuff with heat stress. Sometimes the bad repro or the low preg rate is not happening in July. It's going to happen 2-3 months later, because that's when those primordial follicles are going to be there, and that's how we're going to have that problem. So the same thing here. So we wanted to look into that. Is there any impact in the follicles or the embryos? So one trial that we did at the University of Illinois, we got 72 hosting cows from close-up 30 days before calving until 72 days after calving. And what we did was we fed 4 different diets. A diet that had rumen-protected methionine, rumen-protected choline, both of them, or control. That were our 4 treatments. And mostly I'm going to be talking more about on the methionine treatment here. These were the diets before calving: fresh cows until 30 days in milk and high, and I have nothing to say about it. It's just a really common Midwestern diet. You don't see any almond hulls or stuff that you guys use here. It's just not common for us, or not easy for us to get. Beet pulp, very hard to get, and all this. This is the analysis. Again, just throwing out there our levels of starch, not Wisconsin levels, like 30%. We are pretty much medium-low starch levels in Illinois, and just showing here, this is the common diets we fed them. As expected, what happened when I fed or didn't feed methionine? Protein goes up, milk protein goes up, usually if you're feeding that amino acid that is in need. And milk yield went up as well when you're looking the first 30 days off after calving on those cows. So that's something that we know happens. Now, what we did as well, we followed those cows every 2 days, starting on 7 days, and we are measuring the follicles of those cows. And we say, okay, literature is telling us that cows are going to be ovulating around 16 millimeters follicles. So whenever that follicle from the first follicular wave got to 16 millimeters, we went there and aspirated the follicle fluid. So we had cows that we just followed them, and the other half we aspirated the follicular fluid. And with that, you also get some cells. So we're going to talk about that. One thing that we said, and again, we have a very small n here, so it's more on a, how can I say, on a pilot or on an experimental way, on say, hey, is there an effect? So we said ovulation, how many days it took for them to ovulate for the first time, right? So usually 14, 13 days, no difference there. Growth rate, when I do the millimeters by the days, didn't see anything there. And ovulation size, didn't see anything there statistically as well. But I think this deserves more attention with a larger volume of cows. I think this is there. I think there is something there, and I'm going to show why, especially with the size. This is the diameter, and here I'm doing, so some people would say that really important is those 4 or 5 days before ovulation. That's where it's a kind of exponential growth of that follicle. And you can see that methionine cows were always higher there. So that's how I think it would be interesting to see this with a larger pool, with a larger end, just to see that. And the growth rate, you can see that the cows on the methionine seem to have a little bit more of that growth rate. But let's say, okay, they're all similar. I didn't see any statistical difference. Just saying, I think we should look into that. One thing that you can do when you aspirate, though, that follicular fluid, is that you're getting cells from that follicle. Granulosa cells and theca cells. I didn't have the ability to separate those. So I'm showing you for both of them. We do know some genes that are expressed just in a specific cell. So we could try to figure that out. But I didn't separate them previously to doing the gene expression work. This would be theca cells, granulosa cell, follicular fluid, how things are metabolized, or the pathway of how things are. So I can pick some of these genes and see if they are different expressed in those cows eating methionine or not. One of the things that we were interested in was, of course, in this steroidal kind of pathway. And what we did, some of these receptors for genes, but one of the genes that we found a difference, statistical difference, was this guy here that if you know, that's the one that is pretty much going to be responsible for building up that estradiol inside the follicle. I always have a hard time saying this, but it's the 3-beta hydroxysteroid desaturase. I always have a hard time saying, but that's the one that probably is going to be putting some of my estradiol in that follicular fluid. That's related to follicular size. That's related to more chances of getting that cow pregnant. So we saw that with the cows that with methionine. The other thing, we measured the amino acid concentration inside that follicular fluid. And as expected, methionine had cows with supplementation of methionine, they had more methionine in the follicle. Makes sense. Lysine, we didn't see a difference. And we saw a tendency for histidine. That's the other game that you're going to start seeing, is that if I fulfill one amino acid, now I may need another one to be fulfilled to do the whole nutrition of the cow. So is the histidine the next one? I don't know. That's something that we are trying to look at, but something I'm not going to talk here, we can talk later, but is all this methylation capacity that methionine and choline have, histidine also is involved with that. There's a whole review on how histidine is involved in methylation. But coming back, when I measured the serum of those cows from pre-calving 73, you can see consistently, if I'm feeding rumen-protected methionine on those cows, they are going to get that methionine in the serum as well. How about the uterine health? What we saw is that we can do a pap smear or little brush. We put in the body of the uterus of that cow, and then we put in a slide, we color, and we can count these white blood cells, this polymorphonuclear, and call them PMNs. So we can count them, and there's a lot of literature out there saying, hey, if you have this much, the chances of her getting pregnant is lower, higher. It seems like she has an endometritis. But one thing here that we found, so here you have cows with methionine and cows without methionine, the control. You can see that at 15 days in milk, that PMN is higher, but after, when you go to 30 days or 72 days, that profile switch. And I think this is something that we, maybe as academia and researchers, I think we are going that way now, but we need to do a better job explaining perhaps what is good inflammation, what is bad inflammation. Maybe at some time, especially after calving, where I have perhaps a lot of things inside the uterus, I want to clean the uterus of the cow, higher PMNs or higher white blood cells, that's a good deal. Now, when I try to get this cow pregnant, that's not a good deal, because that may interfere with my semen and everything else. So I think we need to do a better job identify what is good, what is bad, and also it's going to come back to which moment it is. But we believe that by supplying what the cow needs, again, I don't think that rumen-protected methionine is any magic. It's just supplying what the cow needs. That's it. That's our requirement. I don't even call an additive. Some people would call additive. I don't think so. I think it's just a requirement. I think it helps with that. If we go back, there is a very nice paper by LeBlanc here on more on the concept of how inflammation, uterine inflammation on metritis would work. And you can see here that one of the things, and again, this is like, perspective. This is like a model. This is not research yet. That's his idea of it. But these healthy cows here, you see that they overreact in the beginning, but they are able to lower those inflammatory response as lactation goes. However, if you see cows with endometritis, that doesn't happen. They may start a little bit low, but they keep high the whole time. They are not able to lower that PMN. So I think if we are feeding the cow to her requirements, we are going to be able to get more of those healthy cows. How about the embryos? We follow those cows, and at 72, 73 days, we aspirated those embryos. We found some things, but bottom line here that I'm going to show you, that's in a paper there, where actually Anna is one of the authors. What Anna did for us, we just sent that embryo, half of the embryos we sent to Anna in Florida, and she was able to measure here the amount of lipids that we have inside that embryo. That's something I didn't talk about, but oftentimes when we feed methionine to cows, the butter fat or the milk fat is also increased a little bit, and we don't understand really why that happens, but sometimes that happens. Guess what? We saw that the embryos from cows that were fed the methionine had higher lipid inside. Again, I'm not saying that they are fat. I'm not saying that they are toxic fat, and I think we always think about the embryos fat is bad. I think we need to think a little bit different. I'm just saying that it had more than the control. That's it. Interesting enough, 1 year before, work done by Wiltbank and others in Wisconsin medicine, what they did was they fed methionine or not to cows, and they harvest embryos as well. And they did transcriptomics on those embryos. Guess what? Out of the 30 more relevant genes that they found on those transcriptomics, only two were, how can I say that? They were positively higher, or they were higher expressed in those cows fed the rumen protected methionine. Guess which ones were those two? This one's here. One, we don't even know what is it. That's the thing on transcriptomics. You just amplify stuff, and it is what it is. At some point, we're going to figure out. But another one was a polypoprotein. So remember that all those proteins, a polypoprotein, it's involved in the transport of lipids. So all the things responsible for example, and we assume that cows don't have a good physiology for exporting fat from the liver, for example, the VLDL deal. We think that choline helps with that, phosphatidylcholine. This guy here may be helping with that as well, but also can be involved in other things. So the lipid story seems to be now what type of lipid. When we measure it was lipid. Now it's a membrane lipid, it's an internal lipid, it's the signaling that's being affected, we don't know. So one thing that we are doing now, and to be honest, that's something we are working with Purdue, and they have the ability to do this. There's some papers on, they call the lipidomics. So this is an easy way that I have each circle here, I have an embryo, and when you can see here, just like a weather pattern image, you can see how much more or less of that specific lipid you have and where it is. So I can locate where and have an idea on how much. I just got this last week from the other half of the embryos that we had sent to Florida. So we are working on phospholipids, triacylglycerol to try to figure out what it is. Is it something that is in the membrane, is inside, it's being used for storage of energy, or is signaling? That's some of the things we want to figure out. The other thing also with from a colleague here, from Matt Wheeler's lab, one of the things that he did is he's trying to work a lot with this. Not sure if you guys have this, but it's a pretty promising technology, especially on the cost side. So this is very, very cheap. The idea is that you just shaking the molecules, and then throughout this spectrum, you'll figure out what it is. So instead of spending $100 to figure out the amino acids in the plasma of my cow, I could spend $9 to do the same thing. We're not there yet, but I think we're going to be doing a lot of things with this technology, with NMR. And one of the things that the Wheeler's lab did was they measured the amount of amino acids in the media of some cultivated embryos. And this is what they saw. So this is embryos control here, okay? And then you have embryos 1 to 3 days, 3 to 5 days, or 5 to 7 days embryos. And you have control, male or female. My whole point here is, and that's something that we are looking now, that females and male embryos, they behave different. And I think we're getting started to understand that. But you can see that histidine, methionine, and lysine. So this is measuring the concentration of those amino acids in the media. So it's pretty much if the embryo is using, that media is going to go down. If the embryo is not using, it's going to be there. So overall, there's not much activity in the first days of the embryo life. However, as it grows, they really start using more of that. And what it's interesting for me here is the fact of the male-female behave different, but also that seems that methionine is the one that keeps wanting to use the methionine at least, that embryo. If I see lysine or if I see histidine, there's an indication that the embryo is not in need of that anymore. Not for methionine though. So I thought that that was kind of very interesting. Another trial, last one now, also done in a commercial farm in Wisconsin. They fed methionine or not. And here they went from 30 days after calving until 128 days after calving. So they top dressed the methionine or not. So this is how it looked like, the top dress, in usually 20 grams. That's what they did. Yes or no, methionine or not, top dress to cows. They had 309 cows, so they had some primiparous, but they have some multiparous cows as well. Conception rate was all the same, but one of the things that they found different was for primiparous, there was no difference for pregnancy loss when I did at 28 days and then I checked again at 60 days. However, on my multiparous cows, the cows with rumen protect methionine, 6% of pregnancy loss versus 19%. I already told you from the beginning, 19% is not a huge number. We saw farms going, other trials showing from 11 to 20. But this is very interesting. How come they had such a lower pregnancy loss? Coming back to the things that I told you from the beginning, what they did, this is an ultrasound image, and this is the embryo. What they can do is measure the volume of this embryo. Have an idea, again, remember the size is important. And guess what? That's what they, not for the primiparous, but they saw that the multiparous cows, the ones that get the methionine, the embryo was bigger. So probably that was one of the reasons of cows having lower pregnancy loss with the rumen protected methionine group. My idea is, can we say this? I don't have the answers yet, but I think we should be thinking if that embryo at 7 days has more lipid and then it's larger and you have lower pregnancy loss, maybe the idea is that that embryo is just have more energy to grow. That maybe it just has more capacity of exchanging signals and nutrients with the uterus. I don't know yet. That's one of the things we try to understand. So in summary, whenever we feed that rumen protected methionine, it seems to be getting to the follicular fluid. We believe that because of all the primordial follicle and everything else, we should be feeding that before calving. Embryo quality also being affected with that. We believe that's happening. And this would be just some of the kind of overall recommendations or concepts. So for our diets in the Midwest, so you guys can have an idea in a sense, this is how much grams of rumen protected methionine or grams of lysine. That's something that I didn't talk a lot here, but we always try to keep a ratio. NRC would tell you 3-to-1 ratio. We are seeing 2.8, 2.7, it's something better or it's good. So that's what I'm talking about here. And also an idea on that amino acid related to energy also that you're feeding to that cow, it's kind of important as well. But this would be for our diets. Perhaps your diets here, they need more or less. That's the idea. The idea is to feed whatever is needed, not to use rumen protected methionine. Blood meal prices, corn prices, soybean prices, canola meal prices, always going to affect this. If you can formulate a diet minimizing using extra source, you're going to do that for sure. So that's what we believe. And with that, that's what I had for you guys today.
I think we have time for a couple of questions, quick questions.
Why don't you think the primiparous and the multiparous had similar results with the embryo?
I think that's a very good question. And I think what we are getting into is that most of that methionine still, I think, is going through milk. So I think that the demand for milk protein in the multiparous is much higher than in the primiparous. So it would be something similar to why don't we see a lot of hypocalcemia in primiparous cows versus multiparous cows. I think it's just input and output that you're having more on those multiparous cows than the primiparous cows.
Have you thought of labeling the methionine to see where it goes?
Yeah. I see all these labeling papers, and I think they are amazing. But for me, I don't see how I can do that in my capacity. But I think that would be great. I think that would be great. And specifically, I think we are always trying to match that methionine and keep that ratio. We are always feeding lysine, but lysine doesn't seem to show up as something required or provoking a really important pathway or change. But I'm not sure if it's just the way we are looking at. So I don't know if we also label some lysine, if we could see exactly, is it perhaps not so much in the milk, but perhaps more in tissue or something. Yeah.
I find it kind of curious that you were looking at the methionine requirements based solely upon the amount that you fed to the animal rather than talking about optimal levels of methionine in various fluids, including blood, for example, that you could measure. And then you boldly come up at the end and say, well, you should feed 20 grams per day. But what happens if you've got a diet that's already containing a lot of methionine from corn products, for example? I mean, there's a number of studies in the literature that would show negative effects, and we've seen negative effects in studies as well to eating too much methionine.
Yeah, I think that's a very good point, and I think if I would start by the requirement of the cow, and then we can talk about the requirements through NRC model, through Cornell model, through other models, but I think we would have to go through that assessment and feed what is required. So I'm saying here that's what we did for our cow. So for example, two things, the amount and the ratio. So our Midwestern diets, we are usually deficient in methionine, and the ratio is around 3.7 to 1. That's usually what we get in the Midwest. So for our diets, that's what we did. But I would say if the requirement, you're formulating diets for cows, and it's showing you that you are okay in methionine, I see no reason why supplementing it. And I think it's going to be also related to amount of milk being produced. So perhaps cows with lower milk production, the requirement is going to be lower than cows with a higher milk production, for example.
But it would seem to me that you actually have that data already because you've already measured levels of methionine in blood, you've measured it in liquids, and you've also measured the reproductive performance of those animals. So why can't you come up with an optimal blood level of methionine or follicular level? We're above the range.
Yeah, I think that's a good point. My data, what I think I can draw conclusions is if I feed, I have higher. Now, to the exact amount, I think I would have to do a dose response. So for example, when I feed methionine, I got 25 micromolar in the serum. When I don't feed, I had 15. Now, is 25 the best one, or 27, or 22? I don't know. If I did a dose response, perhaps I could have, okay, I have this much methionine, this much, and then I would have probably go back and see, okay, what are the effects that I'm seeing on pregnancy loss and everything else? I think I would have to have a little bit of, how can I say, a little bit more strata to figure out, hey, this is the best way. So for now, I think it's more than 20 micromolar, but I'm totally guessing in the blood. But I think that's some of the things that people are trying to figure that out, to say, hey, if you feed first, is it showing up in the serum? And I think we're going to have to figure that out on what is the best level in the blood, I think.
Okay, so with that, because of the time limit, if you have questions, you can personally ask. Now, let's thank Dr. Cardoso.
Thank you. Thank you.